my interview with Professor Anurag Tandon by Lauren Levy. So I have a couple questions today about your paper on non-invasive gene silencing for α-synuclein. My first question is about the blood-brain barrier. The blood-brain barrier under healthy conditions is a protective layer. I was curious if there's any risk of introducing toxic elements or debris to the brain when the blood-brain barrier permeability is enhanced by focused ultrasound and microbubbles, such as immunogenic antigens.
Yeah, that's actually one of the first questions most people ask, given we know we have a protective layer and we are disrupting it. Now, you just have to remember that in any kind of brain surgery, first of all, the first thing that happens as you cut into the brain, whether it's for epilepsy or cancer or anything else, you're always cutting to the blood-brain barrier. So the blood-brain barrier is always being slightly permeated by any type of neurosurgery. And the goal of our approach is to minimize the length of that time. So in this case, we know that in about 6 to 8 hours, the blood-brain barrier is closed. And this has been done repeatedly in animal studies and more recently in human trials, for example, for Alzheimer's disease, where, as you may know, there is also a risk of vascular amyloidosis in blood vessels of the brain. And so it was a real challenge. The question was to try and address whether those blood vessels in Alzheimer's patients would also react the same. And in fact, they did. In a early trial with 5 patients that had 3 repeated focused ultrasound treatments over several weeks, what they showed was, this was a study done at Sunnybrook with my colleagues. What was shown was that the brain was, the blood-brain barrier was opened, and when the patients came back the following day and were scanned again, it had completely closed over. And the way they do this is using a MRI tracer, which is injected in the bloodstream, which doesn't cross the blood-brain barrier under normal circumstances. And you can see it permeate when the blood-brain barrier is opened. And the day after the sonication is done, the opening is completely closed. Now, the question is related to whether there are, pathogens that might get crossover through an open blood-brain barrier, and that is true. So the risk of having viruses or bacteria that might be bloodborne that can pass into across the blood-brain barrier when it's permeated is not a trivial issue. And so, as in all surgeries, patients have to undergo blood tests and also be assessed for any sort of viral infections they might have. So those are the normal pre-surgery precautions that are taken under any type of surgery. Now, so far in the animal studies, it doesn't appear that they have the blood-brain barrier opening does not seem to induce any deleterious effects. The question is whether repeated openings might, but even there, it seems that as long as you don't open the blood-brain barrier to the extent where, excuse me, large blood cells are also crossing through, it seems to be quite safe. And in some cases, there have been a number of studies that have suggested that it might even be beneficial, for example, in terms of neurogenesis, in terms of improving how the brain reacts to various insults. So I think the long-term prognosis for blood-brain barrier opening, I think, is quite good. It has very limited number of deleterious side effects and a lot of positives in terms of not requiring direct insertion of surgical instruments or any other tools into the brain.
Wow, okay. So actually, out of curiosity, why would it be beneficial? Like,
I'm sorry?
you mentioned that it could be beneficial. Out of curiosity, like, why would it be helpful?
Yeah, we don't. I
Therapeutic.
think that's been shown mechanistically or empirically. We don't know what the rationale for it or what the basis for it is. We do know that there are increased number of new neurons that grow out as neurogenesis seems to be stimulated. There's also the possibility that certain types of factors peripherally may have a beneficial effect in the brain. And again, we don't know what that is. So for example, when you look at treatments of Alzheimer mice, and this is work done in Isabella Baer's lab, showed that when you sonicate animals that have high levels of amyloid in the brain or beta amyloid, you can also treat them with A-beta antibodies. Those antibodies tend to reduce the amount of A-beta accumulation. However, if you use just the sonication alone, there is still a significant benefit from sonication alone relative to the unsonicated hemisphere. And it's not as good as having a sonication and the antibody, but there is clearly a benefit, which suggests that there may even be anti-amyloid antibodies floating around peripherally that are delivered to the brain when the sonication is done.
Wow.
The short answer is we don't know what benefits there may be of opening up the blood-brain barrier. There certainly are risks involved, for example, as you mentioned, from pathogens. But as long as you control for those and make sure that the patient is healthy, it is very likely that those would be trivial issues when you're dealing with clinical delivery of drugs.
So exciting. Okay,
Yeah.
I'm going to move to the next question.
Okay.
So transgenic mice, I noticed that you chose to use transgenic mice with wild-type human α-synuclein rather than mutant α-synuclein. Is there a particular reason for that?
There was no need to use the mutant mice. For example, the A53T mice over time do develop some aggregation, but the mice we chose are lacking the mouse synuclein protein. So they've been knocked out for the murine synuclein gene, and they only express the human gene, which is a transgenic version. So for our purposes, the goal was to really knock down alpha-synuclein levels in general. And this is a proof of principle study where we could target specific parts of the brain. And so there wasn't any need to do any pathological analysis of aggregated synuclein. It was really to see whether the delivery of virus with anti-alpha-synuclein shRNA could knock down synuclein expression in specific parts of the brain. Now, there are a number of other ways to do this study, like you mentioned, using mutant alpha-synuclein. And one of the other studies that we have now underway is, in fact, looking at mice expressing alpha-synuclein A53T mutants. And the rationale for is simply that in this case, we want to rescue the pathology. Now, in the future, it's going to be important to not rely on mutations, because that's a very small part of the human population that actually has PD as a result of altered gene expression or mutations. So our goal in the long run is to be able to target healthy synuclein expression to see whether we can knock that down, because we expect that most idiopathic cases of Parkinson's disease are not related to overexpression or mutations. In fact, most of them would appear to be normal alpha-synuclein that has been altered post-expression in some way. So some sort of post-translational modification and age-related changes that induce synuclein aggregation.
Wow. Okay. That's really cool. Question 3.
Okay.
alpha-synuclein silencing in therapy. So alpha-synuclein is often associated with its potential for toxic gain-of-function aggregation. However, it is also associated with a variety of functions under healthy conditions, and loss of healthy function may also be a toxic pathological feature related to Parkinson's. So I understand that the vector used includes short MIR30 microRNA insertions that reduce toxicity in dopaminergic neurons relative to the shRNA lacking insertions, and that the gene silencing applied in this study was associated with an absence of cell death. Nonetheless, I'm curious, what role would knockdown or gene therapy like this play in therapeutics?
Yeah, that's a great question. And it's one that we don't currently have an answer for because there's a lot of discussion in the literature that knockdown of synuclein might be partly toxic. There are several groups that have looked at changes in immune function, for example, or in the health of certain parts of the brain following synuclein knockdown. Some groups have found that it does seem to cause some toxicity in our hands, and other hands have not shown that. Now, the difference between some of these studies might be related to the extent of the knockdown. So our goal has usually been to reduce synuclein levels by about 50%, so that they still have a sufficient amount to undergo normal, let's say synuclein's involved in vesicle trafficking. Well, we would expect to see the same amount of vesicle trafficking even if it was knocked down by 50%. Those other studies that looked at very, very high knockdown levels of approaching 80 to 90% knockdown suggest that yes, synuclein, if you completely remove it, it will have a deleterious effect. Now, there is a way to get around this. We know if you think about the word alpha-synuclein, you might assume that there are betas and maybe gammas, and then you'd be right. So there's a beta-synuclein and a gamma-synuclein. The gamma-synuclein seems to be mostly associated with peripheral tissues. However, beta-synuclein has a very much brain-specific localization, and we think that it probably will compensate for synuclein knockdown effects if you were to increase it. So, and the reason we would use beta-synuclein also is that it's lacking the sequence that is involved in aggregation. So there is a core sequence in alpha-synuclein that self-assembles, and that is missing in the beta-synuclein. So one possibility would be, as we knock down alpha-synuclein, we would upregulate in the same vector beta-synuclein levels. And that might be one way of getting around the loss of synuclein function. And again, as I say, the loss of function of synuclein may be impactful not just in the brain, but also peripherally. There is some evidence that synuclein plays a role alpha-synuclein plays a role in innate immune function. And so there may be roles of alpha-synuclein that we don't fully appreciate. We tend to think of it as a brain protein, but it is expressed peripherally as well. So my goal would be to show that we can knock it down safely, but also in cases of some sort of toxicity, we would then try and replace it with some beta-synuclein or even a modified version of alpha-synuclein that doesn't self-assemble.
Wow, that's so cool. Okay, number 4.
All right.
So healthy alpha sign versus aggregate alpha sign. As a follow-up question to that regarding both gene silencing and antibody-based therapeutics, can therapeutics like this inhibit toxic alpha-synuclein while maintaining or restoring? I guess that would be resolved by upregulating beta-synuclein. But if so, I guess the considerations would be removing the portion that is involved in aggregation of alpha-synuclein.
Yeah, and that's also a terrific question. Now, the way we've done these studies right now is really been to deliver a viral vector that can knock down alpha-synuclein levels. But as you mentioned, the nanobody studies that we're starting soon would also allow us to target total levels of alpha-synuclein. But if you were to target an antibody, and I don't know if your group is aware of what antibodies are, these are simply very, very small components of antibodies that are composed of just the heavy chain, the variable domain of the heavy chain. So that's the part of the antibody that recognizes an antigen. We can clone these, put them into a viral vector, and then deliver them into the brain to either express intracellularly or extracellularly. And so we have one that can bind to intracellular α-synuclein and deliver it to the proteasome for protein degradation. The basis for this study is simply to look at whether we can knock down total synuclein. Now, at some point, we might be able to target only aggregated synuclein, for example. And so there may be another approach that we can make intracellular nanobodies that target only specific forms that we think are deleterious and remove those. And we could even get more clever about it and put on tags onto our nanobody that only go to specific cell compartments, for example. Let's say we want to target synuclein that accumulates in lysosomes. And we know with age, lysosomal functions tends to be less robust. So we could target these nanobodies to be delivered directly to lysosomes, where they would bind up fibrils or oligomers of alpha-synuclein and then sequester them. They may not destroy them per se, but they would at least be able to sequester them and prevent them from self-assembly. So there are lots of ways to play around with this, and the challenge for us is to find one or more than one that works.
That is so cool.
Yeah.
Okay, I guess final question.
All right.
Just in general, what future directions or challenges do you foresee addressing next with regards to the use of focused ultrasound and α-synuclein in Parkinson's disease?
Yeah, so one of the main challenges for any drug delivery mechanism, and focused ultrasound is a particular one because when we're targeting the brain, one of the main aspects of why focused ultrasound is useful is because it allows us to target a specific part of the brain. Otherwise, you could just inject antibodies into the bloodstream. Maybe less than 0.5% will get at the brain. The problem is when you want to target a part of the brain, the question you can ask is, what part of the brain do you want to target? And so depending on the stage of the disease, from early to middle to late, your targeting may be very, very different. So very early in the disease, and that was one of the rationale for our study, where we targeted the dorsal motor nucleus, the substantia nigra, the hippocampus, the olfactory bulb, because these are all targeted either early or late stages. And so we know we can target them, and we know that we are able to deliver the vector to those particular areas. The problem is once you get into a clinical situation where you have a patient who presents with Parkinson's disease, how do you know which stage they're at? And so we may be required to do imaging studies. And one of the problems currently is that there are no good PET ligands for alpha-synuclein. There are, ways to measure total dopamine storage. So you could do fluorodopa uptake. So there are ways to look at the pathology in a non-invasive way as well. And I think those things will get better over time. So the long-term goal for any Parkinson gene therapy or small molecule therapy and drug delivery will require us to know what stage the patient is at. Can we target that part of the brain effectively so that it prevents pathology from moving to another part of the brain? And I say this is a real technical issue because we don't think we can at the moment rescue cells once they've died. So if you've lost a part of your brain that is involved in a specific function, the neurons in that part of the brain are damaged, we don't have a way of rescuing those and bringing back healthy neurons. There may be a time where we are able to do that. So for example, you might be able to deliver a growth factor that allows a particular cell type to grow. You might then have another factor at a distal part of the brain where you want those new cells to grow out to. You may want to be in, and that's one of the really promising features of focused ultrasound is that you can target different parts of the brain uniquely to deliver drugs to those areas. So in the long run, it would be fantastic if a patient came in, we'd give them the initial gene therapy, they get some growth factors a week later to help not only have you rescued or down-regulated the amount of pathology, but now you've upregulated the amount of growth factor that is involved in that. So those cells start to grow. And then 5 weeks later, as those cells are growing, but not knowing where to project to, you now deliver another stimulus or another vector to some other part of the brain and says, here, come over here. And, this is all science fiction right now, but we don't know where gene therapy is going to be. The vectors we use today are not going to be used in 5 years. They'll be ones that are much more specific, much more targeted. So I think there's, from a technical standpoint, gene therapy is very, very rapidly evolving. I think the drug delivery aspect for using focused ultrasound will become more and more efficient. So currently, the focused ultrasound transducer we use is a single transducer when we use in the animal models. The versions that are being developed for human treatment will have over 1,000 separate transducers for the sonication. That means there are sonications coming from different angles that you could have a very, very much of a focal point. And so as we get more and more precise, you'll find that the regions of the brain that you can target are also more and more precise. And this is a really exciting time for all of that work. So I'm happy to be a part of it, just a little part of it.
This has been incredible. Thank you.
No, you're welcome.